Hello, Daniela. It's my pleasure to welcome you here.
Thank you.
So it's for the prize for the young researcher for the filière Oscar, so it's really a real pleasure to have you here today. So you are coming from Strasbourg, yes, and so we will hear you this afternoon for your talk and your research work on the cytosolic sequestration of the vitamin D receptor as a therapeutic option for vitamin D induced hypercalcemia. And so the floor is yours, Daniela.
Thank you. Thank you for this prize, to giving me the opportunity to be here presenting my work. Vitamin D is a sequesteroid hormone that needs to undergo to a series of hydroxylation steps to be activated. The first one takes place into the liver on the carbon 25, and the second one on the kidney on the carbon first, to produce the 125 dihydroxyvitamin D, that is actually the bioactive form of the hormone. The major biological function of vitamin D is to regulate calcium level. Calcium homeostasis is controlled by a series of hormones in tissue, such as the parathyroid gland, that modulating the production and the secretion of parathyroid hormone, or PTH, will sense the serum calcium level. In particular, when calcium levels are low in the blood, PTG will produce PTH that will increase the hydroxylation of vitamin D into the kidney to produce the bioactive form, that in turn will increase the calcium absorption into the intestine, calcium reabsorption into the kidney, and under certain circumstances, calcium reabsorption from the bones. This will increase calcium into the blood that will shut down the production of PTH. This loop maintains calcium level in a very narrow range. On the other hand, vitamin D can regulate the expression of CYP24A1, that is an hydroxylase that will catabolite and metabolize the vitamin D. This loop will maintain vitamin D in a very physiological levels. Vitamin D, to exert its function, will bind to the ligand binding pocket of its receptor, that is the vitamin D, or VDR, that belongs to the nuclear receptor superfamily. VDR, heterodimerizing with RXR into the nucleus, will bind to specific DNA region called vitamin D responsive element, or VDRE, that is present in the regulatory region of the VDR target gene. Vitamin D function has been studied by the observation of several pathological conditions. For instance, when vitamin D production or activation are inefficient, or in case of loss of function mutation of VDR, we have an hypocalcemic condition. Hypocalcemia is characterized by muscle weakness and especially by rickets that will result in weak and soft bones into the children. These patients are treated with the administration of vitamin D or vitamin D analogs or calcium. On the other hand, when vitamin D levels are above the range, we will have an hypercalcemic condition. Different can be the cause of this or such a condition. Can be, for instance, iatrogenic or accidental vitamin D overdose, some cancer or some syndrome that will produce vitamin D, or especially in the children, mutation, inactivating mutation CYP24A1 are the cause of the idiopathic infantile hypercalcemia. At the moment, there is not an efficient treatment for these patients, especially children, that are treated with diuretics, avoiding calcium and glucocorticoids. But these treatments are not effective due to the severity of the side effect. So, since these patients are characterized by hypercalcemia, hypercalciuria, elevated vitamin D, low PTH, and psychomotor delay, but especially this high level of vitamin D that exerts its function through VDR, we can suggest VDR as a novel therapeutic target. There has been a big interest by the pharmaceutical company to develop and synthesize molecules that are analog of the vitamin D, mimicking the biological function of VDR. But there are few types of VDR antagonists. One of those is ZK. ZK is characterized by a very rigid side chain, and little is known about this compound, and that was studied basically only in vitro. So the in vivo function are not determined. So to this aim, we established a fibroblastic cell line from a patient carrying a mutation of CYP24A1, and we use normal fibroblast as a control. Protein level CYP24A1 was determined only in the normal fibroblast and not in the patient-derived fibroblast, demonstrating that this type of mutation induces protein loss. So we treated this cell line with vitamin D alone or in combination with ZK, and we observed that ZK abrogate the vitamin D induced transcript level of two genes that are present in the skin. So suggesting that indeed this compound is a potent VDR antagonist. Since another target tissue of vitamin D and VDR is the intestine, for the further analysis we use this small intestine cell line that were treated with vitamin D alone or in combination with ZK for different time point. At any time point that we tested, ZK abrogate the effect of vitamin D on the transcriptive level of CYP24A1. To study then the effect of ZK on the calcium flux, or in the cellular calcium flux, we treated the cells, as I said before, and we observed that indeed, after vitamin D treatment, calcium flux was increased, while in the co-treated cells, the calcium flux was the same of the vehicle-treated cells. Different can be the mechanism of the antagonist. So we check the protein level of vitamin D at different VDR at different time point after co-treatment, and we observe that indeed ZK antagonistic activity do not result from the degradation of VDR. I've said before that after the binding of the ligand, VDR has to translocate to the nucleus where it acts on the genome. So we check the effect of ZK on this cellular translocation by subcellular fractionation. We saw that after vitamin D, VDR is mostly located into the nucleus, but after combined treatment is kind of sequestrated into the cytosol. This results was further confirmed in vitro through a collaboration with Natasha Rossell at the IGPMC, that checking what were the region mostly affected by the binding of ZK into the VDR structure, she observed that indeed the most destabilized region by ZK was a region important for the nuclear translocation. So, checking what are the molecular determinants important for the sequestration of VDR into the cytosol, we perform mass spectrometry, and we observe that the most enriched protein into the cytosol after combined treatment compared to vehicle was WPP4. WPP4 is known to be part of the spliceosome, so present only into the nucleus, this from the literature, but through immunofluorescence and subcellular fractionation, we observe that despite its nuclear localization, there is also a cytosolic signal. To study then the importance of WPP4 into the role of, into the ZK antagonism, we knock down the expression into the cells of WPP4. And we observe that VDR was mostly into the nucleus in these cells compared to the control. And also the transcript level of the VDR target gene were increased. On the other hand, in these cells where VWP4 was reduced, we couldn't observe any longer the ZK antagonistic effect on the transcript level of the genes. So we can conclude that VWP4 is essential for the ZK antagonistic activity on VDR activity. So these results prompt us to investigate the in vivo effect of this compound. So we treated mice with for 6 hours with vehicle vitamin D alone or combination with ZK, and the genome-wide analysis was performed on the intestine of such mice. We observed that indeed the 99% of the gene induced by vitamin D were abrogated by ZK, suggesting that indeed also in vivo ZK is a potent VDR antagonist. To test then the therapeutic potency of these molecules in case of vitamin D intoxication, we treated mice for 2 days with supraphysiological doses of vitamin D, followed by 2 days of vitamin D alone or in combination with ZK. And we observed that whereas mice treated with vitamin D alone were hypercalcemic with a suppressed level of PTH, 2 days only treatment of ZK restored the physiological level of serum and the PTH level. So we can conclude that ZK blunts vitamin D-induced gene expression in mice and in fibroblasts, normalizing serum calcium level, PTH level into the mice. But ZK enhances the interaction between VDR and VWP4, and this is important for the nuclear translocation of the receptor. So ZK represents a selective, potent, and safe therapeutic option for the disease caused by chronic or acute hypercalcemia secondary to hypervitaminosis D. So I would like to thank my lab, Daniel Metzger and Gila Verny, for the help during this project, the current lab member and the former, and the collaborator, the IGBMC, and you for your attention.
Thank you very much, Daniela. It's a very nice example of preclinical development for a very potential development for the patients.
I hope.
we really hope to. So you've shown the different diseases for which this therapeutic could be used in the future. So I have first question for you. Did you do any safety analysis in your mice, or did you look at any tissues? Or
Yes, we checked several times, especially for the when I checked for long treatment with the vitamin D and ZK. We didn't observe any side effect, any in all the tissue that we have what we tested, and also the mice look completely fine also into the cage during the treatment. So there is no, at the moment, no.
Okay.
There is no.
Okay. And is there any dose effect? So
yes.
so did you so when you did you intoxicated the drug, the mice with the vitamin D, and then you applied the treatment.
Yes.
So did you do some kind of dose effect?
Yes, it's active also a lower dose. Here, the one that I show was 1 microgram kilo.
Yes.
That is a very high dose for a treatment, especially for this short time. But is also active for a bit longer time with the lower doses. Yes.
Yeah. Okay. And then there is also other putative effect of vitamin, because here we are targeting, the idea is to treat the potential effect of hypercalcemia, calcitriol-induced hypercalcemia. But I guess we don't want to over-treat the mouse. We don't want to have the off-target effect of the vitamin D receptor. So what
I've tested, I've seen that he reached the physiological level, because here I haven't shown, but we also compare the calcium level that we obtain with the mice that don't have VDR, so
Yes.
they are vitamin VDR null, that are hypocalcemic and don't reach hypocalcemia. So really, this kind of compounds doesn't abrogate fully the VDR signaling. Is able to counteract and stop the excess of stimulation that vitamin D can induce in the system without really reaching the physiological levels. Indeed, all the treatment that I've done so far, here I've shown just a part of the experiment that we did, never reach the hypocalcemia, getting mice sick on the other way around. No.
Okay. And so you've shown also so you did RNA-seq on the intestine cells,
yes.
I guess.
These are mice.
These are mice. So did you check in other target tissue where the VDR is expressed?
Yes, the kidney, because all these things were compared also in the one shot, in the 6-hour treatment, but also in the longer treatment, the one that I've shown at the end. And no, is able to abrogate and is effective in all the tissue that we tested, that is the kidney, the second one. That, yes, is effective also there and is a really potent VDR antagonist, getting the physiological level of the transcript level that are increased after stimulation with vitamin D.
Okay. So thank you very much, Daniela, for
You're
your
welcome.
very clear presentation and answers.
Thank you.
Congratulations for your prize.
Thank you.
So I'm inviting now Arnaud Peramo, who's our... Ah oui, maintenant, oui, pardon, je peux passer en français, excuse-moi Arnaud. Alors, j'invite maintenant Arnaud Peramo, qui est chargé de mission recherche pour la filière Oscar et qui va remettre les Oscars de la recherche, du prix recherche pour nos 2 lauréats. Donc, Joanne, je vous invite à nous rejoindre. Voilà. Arnaud, je te... confie, voilà. Attends, je te donne le micro. Ah, non, mais c'est un vrai. Non, mais il faut le sortir. Il faut le sortir de sa boîte. Hein, il faut le sortir de sa boîte pour le montrer. Hein.
Il finit le petit paquet Oscar aussi. Et pour Joanne, pareil.
Voilà.
Et du coup, au nom du groupe de recherche, on vous remercie pour votre implication dans les maladies rares et pour vos travaux de recherche.
Voilà. Et bravo. Et on espère que vous allez continuer à travailler dans le domaine parce que c'est extrêmement important.
On a besoin de chercheurs brillants qui travaillent sur les maladies rares osseuses.
Merci. Voilà, donc je vous remercie beaucoup d'être resté avec nous jusqu'au bout de cette journée recherche qui était en 3 parties, parfois un petit peu avec les petits aléas techniques. Mais c'était extrêmement intéressant. On a eu des communications passionnantes, du fondamental jusqu'aux applications cliniques. Bravo aux lauréats des prix recherche. Et je vous donne donc rendez-vous le jeudi 25 novembre pour la journée de la filière. Je vous souhaite à tous un très bel été.